So now we're going to get a little bit into regulation of prokaryotic gene expression. And the main model we're going to use is the operon model. And so the first slide here has good information for you to know, and we will go through all of this. So let's start off by considering three cultures, all of E. coli, the same exact strain. The only difference between them is that here we've got growth only in the blue, growth only in glucose. So it's the only carbon source that it can use, the only sugar. The green one is with only lactose. And you can see that, so a general curve, for example, general growth curve looks something like this. And this is on the x-axis, this is time. And then the y-axis, for example, is OD at 600. And so, for example, you can imagine a tube of bacteria over time, same tube, over time, where it starts out pretty transparent. There's a few bacteria in there. And then as time moves along, the cultures get thicker and thicker and thicker because there's more bacteria growing in there, right? And so by measuring the optical density at 600, you're measuring how much bacteria is in the tube, and that's what we're seeing here. Okay, and so you can also plot at bacterial cell density in terms of cells per milliliter if you know those volumes. So you can, for example, take the liquid out of here, and then you could put it onto a petri dish, and then the number of colonies you get, you could then count the number of cells. Or you can actually look at this under the microscope. So this right here, there's a certain period of time that's called the lag phase. And that is whatever the bacteria stage was in, it takes them a little while to start really dividing. And then this is a log phase. And so they're rapidly dividing. Every cell is dividing into two very rapidly. This is the stationary phase. And then this is the death phase. All right, so it's an old culture. There's toxins. It's used up the nutrients and released all these waste products, and maybe the pH is changing, and so the bacteria start dying. So this is, generally speaking, a general growth curve. So if we look over here then, we see the difference between the blue and the green is that it takes a little bit longer for the bacteria to start growing in lactose. And that's because it needs to turn some genes on. So there's a little bit of a lag before it gets to that log phase if you compared these two. Otherwise, it's just shifted. Now, if you look at glucose and lactose in the same tube, what we see is that a curve starts which is remarkably similar to glucose. Now, it's used up all the glucose. There's another lag phase, right? Oh, okay, so there's another lag phase there, which we didn't see before. And then it starts growing up again. So what happens, to explain this, which we'll discuss, is that it's using glucose here. So it's using glucose. It's run out of glucose, so there's that lag phase. And then it's starting to use lactose. In other words, it's not going to use lactose to begin with. So, because otherwise, why not just have this go, ooh, right? If it use lactose and glucose at the same time. Well, lactose is a disaccharide of glucose and galactose. And so you need to cleave that. And so if you've got glucose around already, why make the enzyme that cleaves that? And lactose needs to get into the cell. Why make a permease for lactose if there's nothing present? So that's it. Some bacteria live all their lives without seeing lactose. So why make proteins? Why express genes that are involved in lactose metabolism? So this growth we see here is called dioxic growth, right? Two kinds of growth. So let's talk about how the lac operon explains that. And so in general, what we're looking at here is the general topic of constitutive versus regulated gene expression. So the glucose could get right into the cell, which means that there's glucose transporters in the cell. It gets right in. So the genes for the glucose transporter are constitutively expressed. They're expressed all the time. whereas the genes for lactose utilization are regulated. And so, when we talk about heat shock, for example, when we are transforming bacteria, a whole bunch of genes get turned on. It's regulated. Okay, those genes are not needed if it is in more suitable temperature. Okay, so if we look on here, then both types of expression rely on certain genetic sequences. And if we look at this is a particular regulatory site. And as soon as you see a diamond in the middle, automatically think what? What should you think? Palindrome. And these are color coordinated to show little palindromes in the overall palindrome. So the blue sequence, right? This is the same thing as this, right? This sequence is the same as this sequence. And so this overall sequence is a palindrome, although the black ones are showing where they're different. Okay, but overall it's a palindrome. So we would expect a dimer to bind here. Okay, so repressors prevent transcription of repressors prevent transcription of negatively regulated genes, and activators activate positively regulated genes. So let's look at some of the players. So first of all, these are different regulatory proteins. And if you look at this, see the symmetry in all of these? So those are dimers. If we see a lot of these will have helices, which is the recognition helix, which will go into that major groove, whereas the first helix is often the backbone. And we discussed, for example, some backbone binding of the DNA through the phosphates. And so all of those contacts that we discussed with the three-dimensional molecular landscape, think of it in that perspective. And so again, this right here is a helix-turn-helix motif. And so, we're going to talk about these two, the Lac repressor and CAP. Okay? And then there are some DNA binding proteins which actually lay beta strands into the major groove. So operons is the main topic. And so this is a coordinated unit of expressed genes, and the expressed genes are all on one transcript. Okay, so for example, in this particular example, this is going to be expressed as one polycistronic messenger RNA that has all three of those genes, but then it will be translated at different start codons. So I've color coordinated these, so green, etc., etc. So there's a regulator gene, which encodes for a protein, which will bind to the operator. This will encode a protein, which binds to the operator. There's a promoter site, which binds DNA RNA polymerase. And then there are structural genes. Polycistronic means that one messenger RNA will contain all three transcripts. So here it is again. And so now specifically what we're looking at for these structural genes is beta-galactosidase, lactose permease, and transacetylase. Let's not worry about transacetylase for this class. So E. coli, for the lactose to get in, it needs galactoside permease. And so that's what this is. It's also called lactose permease. These are the same things. So without this protein, this is not getting in. Once it gets in, to break the glucose from the galactose, you need beta-galactosidase. And that's this enzyme right here. So you see, this transcript is going to encode for both the protein that allows the lactose to get into the cell and the protein that will cleave lactose into glucose and galactose. If there is no lactose, why express these proteins? There's no reason to. So this other regulatory gene encodes for the lactose repressor. Now this is going to encode a protein which is going to bind to this operator. See that right there? Lactose repressor binding site. So this is going to go over there and bind to that. Now beta-galactosidase cleaves lactose into both galactose and glucose, okay? But it also has this little side reaction here. And so a very small percentage of product is instead a reconfigured lactose, allolactose. And it turns out that this allolactose is used in conjunction with the lactose repressor protein. So we will talk about that. So anytime we talk about lactose binding to the lactose repressor, it's actually allolactose. So this is a general kind of scheme. The LAC-I is expressed constitutively. Okay. So LAC-I is one that is expressed constitutively with this. So LAC-I is expressed. So this forms a tetramer, which then binds to the operator. When it's bound to the operator, RNA polymerase, which is this, cannot bind. And so these genes are not expressed. However, if there is lactose present, and remember it's really allolactose, that's what this is. So this equals, may as well get a little bit of color coding here, that. And so that right there is allolactose. Well, allolactose will bind to all four of these, and then it's released. So in other words, in this case, allolactose bound here, 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 and here, and then it is released, and that's what we're seeing down here. So it is inducing, allolactose is binding to the LAC repressor, causing it to release the DNA. That allows RNA polymerase to move along, synthesize this messenger RNA, and then on top of this, of course, we will have ribosomes bind this, right? That's my bad ribosome drawing, to produce these particular proteins. So once that happens, then the cell will start increasing the concentration of a bunch of lactose permeases because it's making a whole bunch. There's going to be a lot of beta-galactosidase in here as well, which means that the lactose is now going to be able to get into the cell very easily, and this is going to cleave it to glucose and galactose. So this is showing its negative control because in the typical situation without lactose, it is negatively expressed. It is not expressed at all. Plus lactose, it binds to this constitutively expressed protein, which normally is bound to the operator, causes it to be released, and then RNA polymerase can bind. Right? It couldn't bind here when the LAC repressor was there, and it moves along, transcribes this messenger RNA, and then the ribosomes will bind and then synthesize the proteins. But that's not all there is. So this right here is what we just looked at, the LAC repressor. So it really is a lactose sensor, right? You can get that because it's active, right? Binding to DNA when there's no lactose. And when there is lactose, it's released. And so all this information is important, and this is what we just covered up here. That's not the end of the story, though, because this is also part of catabolite repression. So enzymes that, pathways that are involved breaking down molecules have a general response. So the catabolite activating protein, aka CAP, it's also called the cyclic AMP receptor protein, CERP, it's a glucose sensor. So this right here is a global regulator that tells the cell, is there glucose present? And so that is particularly important because glucose is its main sugar. So by knowing if there's glucose present, that tells the cell a lot of things in terms of the metabolism it undergoes. So it's used in a lot of operons. And so this is the deal. When there's no cyclic AMP bond, the dimer does not bind DNA. So this is different than this. So remember up here, the LAC repressor is bound when there's no lactose present, when it's not bound. And when the lactose binds, the allolactose, it reverses. Well, CAP only binds to the DNA when it's got cyclic AMP bound to it. And when it's bound to it, it actually recruits the RNA polymerase to start transcription. And so let's look at this a little bit more closely. First of all, the fact that we've got this drawn like this means that this functions as what? 1, 2, homodimer. And so this whole business of it being a glucose sensor, well, let's see how that works. So there's this enzyme called adenylyl cyclase, and when it is active, it converts ATP into cyclic AMP. And quick, what's this? What is this? That is, right, when you've got those 2-phosphate linked, that was that was hydrolyzed off, right? That got kicked off. What is that? Oops. Right? PPI, pyrophosphate, right? That's what this is, pyrophosphate. And so you got cyclic AMP. Cyclic AMP is a wonderful second messenger. That tells the cell a lot. In this case, this happens mostly when there's low glucose, because high glucose inhibits this enzyme. So in high glucose, adenylyl cyclase is inactivated, which means that there's low cyclic AMP because this is not working as much. So low cyclic AMP, CAP is not going to be bound with cyclic AMP. So in this case, it does not bind to DNA, and it does not recruit RNA polymerase. Remember this? It recruits RNA polymerase when it's bound. When there's cyclic AMP bound, it recruits RNA polymerase and transcribes. So when we look at this, here's the dimer of CAP, one subunit, another subunit bound to DNA. And look at that, it's binding DNA. That's a common theme as we're seeing. This is another representation showing the two monomers, and what we see right here is cyclic AMP. So there's the cyclic AMP. What we see is that helix in that major groove, right? And so it is then bending it. And same thing over here, but now what we're looking at is we're looking at a carboxy terminal domain of RNA polymerase. So check this out. Let's go over here. This is now showing the dimer of CAP bound to cyclic AMP, and since it's bound to cyclic AMP, it is bound to DNA. And so the DNA would keep going, right? This is just showing a little bit of DNA, but what we see here in the green is RNA polymerase. And specifically what we're seeing here is the RNA polymerase in green, the carboxy terminal domain of the RNA polymerase. That's what the green part is. The whole thing is RNA polymerase. And you see how it's interacting with CAP? That's what's showing here. So RNA polymerase right here, interaction with CAP that's got cyclic AMP bound, that's what we have here. And so this right here is RNA polymerase. And this is just a diagram that is showing, so this whole thing right here is RNA polymerase bound to DNA. And look at that, carboxy terminal domain interacting with the CAP. Carboxy terminal domain interacting with the CAP. That's what we're seeing here. And so when we look at this, what we see is that there are different types of promoters which we don't need to get into. But in both of these, the CAP binds between 40 and 60 base pairs upstream of the start site. So this is the beginning of the gene. Anytime you see this arrow, right? So when you see this, that means that that's where it's going to start transcription. And so what we see here then is that the CAP, which is a dimer, is binding to a regulatory sequence, and this is the regulatory sequence. Tell me something that is unsurprising about this regulatory sequence. That's right, it's a palindrome. And then what we're seeing with these different colors, these different ovals, those are particular sites where the protein is interacting with it. And if you look here, see this GT and see this TG? See this C and this C? See this G and this G? Mirror image, homodimer binding. So this right here is showing CAP bound to the DNA. And again, this is used in hundreds of promoters, right? This is just one of them. So now let's talk a little bit more about that lac repressor protein that we talked about before, that the diagram showed, that there's 4. So each subunit, look at this. At least 3 different domains, and actually this sometimes we'll call that a domain, but really this carboxy terminal alpha helix. So they're in domains. So there is a helix turned helix N-terminal headpiece that binds the operator in the major groove. So that's what's binding it. Then there's a linker which allows this to move around. And then this is the inducer binding site. What's the inducer for the lac repressor protein? What binds to the lac repressor protein in the natural system? That's right, allolactose. And so remember, this operates as a tetramer. And so this tetramer is shown over here. So it's actually a dimer of dimers. So do you see over here, we've got one of the monomers in green and another one in gold. Over here, we've got one in yellow and one in blue. So this is a dimer and this is a dimer. Oh, and check this out. Woohoo! Right? This C-terminal alpha helix on all of them down here, that's what forms a tetramer. Very cool. That's what forms a tetramer. And then you've got this dimer over here and this dimer over here. This is from a crystal structure showing it bound to DNA. Now this image shows two pieces of DNA, but really that's because that's how they had to get the crystal structure. In real life, it's one piece of DNA, but they're not close. Okay, so what does that mean? Well, let's check this out. What we're looking at here is, let's make this a little bit bigger. This is a great website. So this is showing the DNA and one dimer of dimers, just the important parts. What happens when it binds and then it bends that DNA. And oh, look, I can, oh yeah, move it around. So there's a couple things I want to keep your eye on. First of all, right, this is the helix-turned helix head terminal domain. And then remember there's a linker. So look what happens to the linker. Disordered helix. Disordered helix. So when it first binds to DNA, it's bound to DNA non-specifically, and when it binds to the right site, the right sequence, that changes shape and that helix is formed and it gets inside that groove. And so it changes from disordered to the groove. And so we can see that right there. And so this is this right, and actually this is the minor groove, and you see how that minor groove is essentially pried open. Ah, so cool. So the other thing I want to, and now this is a good web page. This has it's a good review of everything we're talking about right now. So the other thing I want to bring is I mentioned kinks in DNA, right? Remember when you have base pairs that are not completely parallel for their base stacking. And so what we can do is we can zoom in on a region that shows, okay, so if you look at this, see how this base pair is parallel with that base pair and there's stacking, same with over here. Well, look at this, right? These are not parallel anymore. That's because the DNA was just bent, and so you don't have that pi stacking anymore, right? However, so you may think, okay, well that destabilizes a little bit, and ding, 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 you're absolutely correct. However, it does, what we have is these two leucines, which is a little weird for this one, but there's two leucines here, which here they are. They essentially get into there, and they are sticking in. If this was space filling, ideally you'd see that it's in there, but they are now in van der Waals contact with this bases that used to be pi-pi interactions. They used to be parallel, but they got pried open. And I'm going to refresh this so we can just remind ourselves of what that looked like, right, when it goes in. And so it bends that DNA. Now not all of the base pairs are going to be pi-pi stacking, and leucines get in there to hold it into place and to stabilize it. So cool. Okay, so you saw some changes for sure. Okay, so let's go over this schematic again. So the lac operon, allolactose is the inducer. When there's no lactose present, then the repressor, lac repressor, is created, and it binds to the operator. This is a very crude diagram. We know that really it's a tetramer, right? And so on that note, I can just maybe draw something like this to show that there's 4 of them there. Okay, although we will see that that's still not quite right. But for the sake of this discussion, so it binds there. And then what we have essentially is RNA polymerase cannot induce transcription. When allolactose is present, it binds to the lac repressor, and it no longer is bound to the operator, and then you get transcription. Okay. And then there's cap, which we've talked about. So now let's talk about a little bit more about the structure of cap. Excuse me, we already talked about cap, we're talking about the lac repressor. So we're adding to this. You saw part of this before. So it actually turns out that there are two operators, operator 1 and operator 2, and that's where each lac repressor dimer binds to. One binds to O1 and one binds to O3. Right? This is a dimer and it's a dimer. So it's a dimer of dimers. And so this right here, if you look at this, this operator is about 500 base pairs away from this operator, so it loops this DNA because these are physically close together. So check that out. It loops the DNA, and then cap is in there. Okay? And so when we look at this particular example, this is ready to go so that if this binds to lactose and is released, cap is there, and as long as glucose is there, cap is there, and then it will go ahead and induce transcription. Now, check this out. Lac repressor binds to the lac operon. It's K, right? About 10 to the 10 molar per second. Whereas general diffusion is only 10 to the 7 molar per second. You remember this number from last semester. So how can that be? It's that whole binding non-specifically and sliding around. It's not working in a three-dimensional landscape anymore. It's sliding along. And so to add the final piece on this, although we've been looking at this is a little bit better representation of it. So the circles are representing the same general area. And what we see is that inside the lac Z is actually this O2. So O2 is actually somewhere around here. And so that means that essentially, right, this is O2. When lac repressor binds, it will bind here and it will bind there. Right? Excuse me, that was supposed to be O3. Goodness. O3. Right? So that's what we're looking at here. And so to bind there and there, that's too far of a distance without the DNA in the middle looping. So this DNA in the middle here will loop, bringing these two operators closer together, O1 and O3, when the lactose repressor is binding it. And so when we look at the promoter, right, that's where the operator, that's where RNA polymerase binds to the promoter. So that's what we're seeing here, RNA polymerase binding to cap on its sequence. Cap sequence and RNA polymerase right here. And then of course over here this produces the lac repressor. So get a good idea of how all this is worked together. Don't worry about O2. Wah, wah, wah. We got enough on our plate, so we don't need to worry about that. And the sequence of lac operator O1, no surprise, it is a blank. So this is the same thing that we've been talking about. Go through this yourself. Make sure that you agree. Yes, this is exactly what we've been talking about. So that explains why you get this dioxic growth, right? At the beginning, when glucose and lactose are present, right? High glucose means that there is low what? It's going to inhibit an enzyme. It's going to hit adenylyl cyclase. So there will be low cyclic AMP. If there is low cyclic AMP, cap is not bound, so will not recruit RNA polymerase. So in other words, the lactose operon is not expressed. Even though at this point, the lactose, the allolactose, is bound to the repressor, so it is not bound to DNA, but still, if cap is not there, you're not going to get transcription. Okay, so that's what happens. That's what happens when you have glucose and lactose. When you have glucose, let's see, lac is bound to repressor. In the absence, that's correct. That's for 1. This is for 2 and 3. This right here, right? Because lactose, you need to have lactose present, so 2 and 3. This is only present when there's glucose, right? So this is going on when you have what? when you have 1 and 3. Both of those have glucose. So that's the situation here. That's the molecular situation, right? So by using that information, what we discussed, you should be able to, this would be a good test question, explain what proteins have what bound to them and what proteins are bound to what DNA elements in every example here and at every part of the curve, right? What genes are being expressed here, here, and here, etc. So tie this up with the molecular the molecular situation, and this is a good topic for us to discuss during a review session as well. Okay, so that is the lactose operon.